Hello everyone, in this video we're going to talk about mechanisms behind speciation. So speciation is the process of having a new species diverging from a common ancestor. So you have your common ancestor, and divergent evolution allowed different species to evolve from the original common ancestor. So there are two ways that speciation can occur. One is called allopatric speciation, and the other one is called sympatric speciation. So if you think about the word sim, means there's something happening at the same time, right? At the same location or whatever. So in this case, the difference between allopatric speciation and sympatric speciation is that sympatric doesn't have a separation in location. All of the organisms or two populations are at the same area when the speciation event occurs. And for allopatric speciation, the speciation process occurs because the two populations are being separated by a geographical barrier usually. So if you look at this picture right here, we have our original population. During allopatric speciation, the middle part of the water kind of, went up a little bit, so which is something that naturally happens within our ocean. So in the ocean, sometimes mountains can come out of nowhere because of the movement of the tectonic plates, I suppose. And so if there's a geographical barrier that comes up between two populations, that's going to separate the two populations and prevent gene flow from happening. And without gene flow, there could be natural selection and mutations and genetic drift that happen to each population on its own. And those the accumulation of those changes may or may not allow the two populations to become two different species. And if they do become two different species, we'll call it allopatric speciation. Sympatric speciation happens when the original population becomes two different species without the geographical, what's wrong with my hair? Without the geographical barrier. So there's still gene flow that can potentially happen, but for some reason, there's enough changes between certain individuals within the population that they eventually become two different species, that they can no longer have gene flow even though they're in the same area. So moving on, in allopatric speciation, here's their definition. Gene flow is interrupted or reduced when a population is divided into two geographical isolated subpopulations. So if you look at your notes, the first picture of the horse and the unicorn, they're separated by this river, right? So that's just a random cartoon, but it demonstrates how allopatric speciation can happen when there's a geographical barrier appearing between two populations. Another example could be at the Galapagos Island. We could have birds at different islands, but then eventually they become flightless, that they can no longer allow the gene flow, so that they are accumulating their own mutations and changes. So the definition of a barrier, when you think about a barrier, you can think of a mountain or a river, anything like that. But for different types of organisms, the barrier could be large or small, and it could be different types of barrier. A barrier for one type of organism that may lead to allopatric speciation might not be a problem for another type of organism. So for example, a canyon might be a barrier for small rodents, but it might not be a problem for birds, coyotes, or pollen. And a lot of the times for plants, the barriers, it's more difficult to have a geographical barrier for plants because the wind can carry pollen for a long distance. So here's a really important part that you should write down on your notes. What exactly could happen to the two populations that are separated by the geographical barrier? So you have these two populations. What could happen to them that actually allow the two populations to become two different species? So as I've said earlier, there could be mutations, right? Because mutations happen to all the individuals within the population. So mutations can obviously happen to the two separate populations. Natural selection. So natural selection acting on the phenotypes that are caused by the mutations could cause a change in allele frequencies, right? So disrupting the Hardy-Weinberg equilibrium, natural selection can cause a change in the distribution of the alleles as well as the distribution of the phenotypes. The last part is genetic drift, because natural selection is not the only way that allele frequencies or phenotypic frequencies can change. Genetic drift, there are random events that can happen, such as bottleneck effect and founder effect and just random gene flow, those could also cause a change within the population. So the second one is important. So reproductive isolation, all those pre-zygotic barriers and post-zygotic, pre and post-zygotic isolations is what we call them. Those could result from all those mutation, natural selection, genetic drift that happen to the two populations, but it's not necessarily, going to happen. It's only a byproduct that may or may not happen. So if you read this sentence, reproductive isolation may arise as a byproduct of the genetic divergence, but it's also possible that the two populations are going to remain the same species. They might not have all those possible, behavioral isolations or temporal isolations or hybrid breakdown. All those problems may or may not actually occur. So for example, there are many examples, but this one gives you an example of mosquito fish that have become reproductively isolated as a result of their own natural selection by the predators. So in this case, this is an experiment done, some observations on these fish. So they're called whatever. Mosquito fish, that's right. They're called mosquito fish because they eat mosquitoes, I think. So there are two species of mosquito fish. One, we can observe them in ponds that have a lot of predators. So there's one pond that has a lot of predators. And then there's this other pond that doesn't have a lot of predators, if you look on the top. So when we look at these two species of fish, we can see that there's a difference in the phenotype. The fish from the pond without the predators are kind of more slim, and the one from the pond with a lot of predators are kind of, bigger and stronger and swim faster, which makes sense. So how would this work? Let's say we have one pond, and then somehow this pond got separated into a pond and another pond. And this pond happened to have a lot of predators. It just so happened. So this pond is going to have a variation or genetic variation or phenotypic variation of fish that looks like this, that can swim fast but are bigger, or this one that's more slim but for steady swimming. So in this pond that has more predators, we'll just call it orange fish. The orange fish are going to have a selective advantage, right? They have the ability to survive and reproduce under the condition of having those predators. So more of those orange fish are going to survive and reproduce, and eventually they're going to become the majority of the population within this one pond. And eventually, through this natural selection process of the existing mutations and phenotype, notice I said existing mutations and phenotype, right? This body shape did not occur because of the predators. This body shape was there in the first place, but it just went through the process of natural selection to make it more prevalent within the population. So I hope that makes sense. And if it doesn't, please ask me at some point. So if you look at the bottom graph, it shows you, just some data that the scientists have collected. On the left side, we have the escape acceleration. So this shows you the phenotype and the actual result of, of having a specific phenotype. So the orange fish, which came from the pond with the predators, escape a lot faster than the fish from the pond with no predators. And then the next one is called, the next one is on the survival rate with the predator. So if we put these fish from the pond with the predators, which in this case, the orange fish are the most prevalent, those fish are much better at surviving with the predators, and the green fish are not as good as surviving with the predators. So what is the point, right? This one shows you that there's a difference in escape acceleration and survival, but what's the point of us talking about this? This shows you that through allopatric speciation of having a certain environment at one place versus the other place for the two populations, this could help accumulating the changes in phenotypic differences that eventually these two fish from these two populations might become two different species, and in this case, they can become two different species. So there are a lot of evidence of allopatric speciation as well. Obviously, the Galapagos Island has all of the evidence for all the types of evolution, but in this case, let's see. But in this case, we can actually reinvent the wheel and kind of do the allopatric speciation process within our labs as as an evidence that can happen right in front of your eyes. So that's basically what this page talks about. So here's another experiment showing you how exactly this works, or this could work within a lab. So we have an initial population of fruit flies, which are called Drosophila, and this one population of fruit flies are separated and raised at two different environment. We have fruit flies raised on starch and some fruit flies raised on maltose, which is a different type of sugar. And what we find out is that we're doing matings between the male and female, and we find out that the female fruit flies raised on starch medium, for the most part, for the majority, they would select males that are also raised on the starch medium, and the females raised on maltose, which is right here, would for the most part select males that are also raised on maltose. And then this one is our control group, where all the males and females are raised on starch, and there's no difference within the selection between male and female. So what does this actually tell you? This shows you that depending on the environment, there's a selection process, right? So the females raised on the starch are going to be better at digesting starch, and they would prefer male that can also digest starch, because then their offspring can survive and reproduce and digest starch, right? And survive and reproduce even more. So that makes sense. Same case with the maltose and maltose. So this shows you, we're kind of reinventing the process of allopatric speciation. Eventually, as you can see, there's already a difference, not necessarily in the trait, that, it's not showing you that there's a trait difference between the male and the female, but there has to be some sort of phenotypic differences between the starch-eating male and starch-eating female versus the maltose-eating male and maltose-eating female, that they're able to recognize each other to allow, the selection of the other mate and continue on the process of survival and reproduction. I hope this kind of makes sense. All right, the next one, obviously allopatric speciation can also be observed in nature. And a typical example is the formation of the Isthmus of Panama of snapping shrimps. So how this worked was 3 to 9 million years ago, the Isthmus of Panama was actually not there. It was under the ocean. So these shrimps that were living in the Atlantic Ocean and the Pacific Ocean, well, they were able to have their gene flow because it was all one part. There was no barrier in between the two populations. However, this Isthmus of Panama started rising and rising and rising from the bottom of the ocean, and eventually separating the two parts of the ocean, the Atlantic and the Pacific Ocean. And the result is, because of the accumulation of mutations and natural selection and genetic drift, all of those factors working together, we now have different species of both types of snapping shrimps. So those two snapping shrimps are, what is called snapping shrimps. So these two types of snapping shrimps are sister species. They came from the same common ancestor, but now there are two different species, and these two as well. So this is a natural example of allopatric speciation. Regions with many geographical barriers typically have more species than regions with fewer barriers, right? That makes sense, because the barriers prevent gene flow, which helps the build-up of the differences between different populations, which may lead to what we call reproductive isolations, which may lead to speciation. And then the second one also should make sense. If two populations are farther apart, generally it will be more likely for the two populations to evolve the reproductive isolation. Okay, so this one right here, reproductive barriers are intrinsic to the organisms. That means, like we said earlier, just having the physical separation is not necessarily going to cause enough biological, well, just having the physical separation is not going to cause the biological barrier, right? So when we talk about biological barrier, those are the prezygotic and postzygotic isolations, the behavioral isolation, temporal isolation, geographical isolation. Those are all, kind of results of our initial physical separation. And then eventually, because of this physical separation, there could be changes biologically, mutations, and natural selection and genetic drift. We said that way too many times, but now you're going to remember it, I hope. Those factors are going to accumulate and actually cause the reproductive differences or reproductive barriers. So now these two populations can no longer have viable, fertile offsprings, which makes them two different species. So now we're on to Sympatric speciation. Sympatric speciation occur in populations that live in the same geographical area. However, so there has to be a reduce of gene flow in order to, allow those mutations and all three factors to actually accumulate. But there are three possibilities on why this could happen within an area with multiple populations with no geographical barrier. So here are the three possible factors. The first one is called polyploidy. The second one is called sexual selection. And the third one is habitat differentiation. So we'll talk about each one of those. We're going to start with polyploidy. So polyploidy means, so you know what haploid means. A haploid cell will have one set of chromosomes. Each type of chromosome will code for specific genes and, alleles and certain traits and proteins. That's haploid. And when you have a diploid cell, such as all your somatic cells in your body, those cells, or as long as they're not mutated, those somatic cells are diploid, meaning that we have for each type of chromosomes, we have two sets. So instead of 23 chromosomes, we will have 23 pairs of chromosomes, and within each pair of homologous chromosomes, you will have the same, genes and traits or alleles being coded for on each homologous chromosome. So that's diploid. Polyploid just means it's not haploid, it's not diploid, it's more than two. So instead of having two sets of chromosomes, you might have three sets or four sets. So instead of big A, little a, as the genotype for a person, you might have big A, little a, and A dash, for example. That will make it a triploid, or a polyploid. So this can happen in the presence, this can happen as an accident during cell division. So if you think about cell division where, let's say mitosis is supposed to have sister chromatids being pulled apart, right? If the sister chromatids are not pulled apart, you're going to have a cell with an extra, sister chromatid. But this polyploidy process, for the most part, happened to meiosis. If you think about meiosis 1, where the homologous chromosome is supposed to be separated, or meiosis 2, where the sister chromatid is supposed to be separated, if that process doesn't happen, or this is called non-disjunction, your daughter cells are going to have more than the normal amount of sets of chromosomes. And this will result in polyploidy. So this process happens a lot more often in plants and animals. I suppose there's less, checkpoints or mechanisms to prevent non-disjunction from happening within plants. Okay, so this polyploidy, let's say you have a plant that's supposed to be 2N, but then because of non-disjunction, we have offspring plants that are 3N or 4N or even 5N. Those polyploid plants could potentially become its own species. So that's what this is about, okay? We can have these plants all living in the same area, but because of these errors happening during cell division, this could lead to new species. So here's an example of how polyploidy can happen. There's autopolyploidy, when an individual with more than 2 chromosome sets derived from a single species. So you would have one species of plants, but because of the errors during cell division, it becomes a polyploid. And the offspring resulting, so this polyploid offspring, from mating between polyploids and diploids have reduced fertility. So this is our reduced fertility. Our F1 generation might not be, or is not going to be, be fertile. So you don't really need to know autopolyploid, this word specifically, but just have the idea that, you know, these are things that could happen to plant, and make sure that you understand how meiosis can happen, and if there is an error in either meiosis 1 or meiosis 2, you should be able to predict what's going to happen to the chromosome number in the end. So this is a good time to review your meiosis. The meiosis is definitely too basic. You have to know it. So here's to show you how this works. So it says the offspring resulting from mating between polyploids and diploids have reduced fertility. So what does this mean? We have an original population, original cell that's supposed to be diploid, and the 2n number equals to 6, meaning that there are 6 separate chromosomes, but we have 2 sets of each type of chromosome. So in total, there are 2 sets of 3s. And if there's a cell division error, so what is this? So let's say this is supposed to be meiosis, but let's say these homologous chromosomes were never separated, then you could have a tetrapolyploid cell, right? Yeah. So if you have this homologous chromosomes not separated, but then the sister chromatid. Wait. So let me think about this for a second. Okay, so what's happening here is that between this diploid and tetraploid cell, this should be meiosis. This should be mitosis. So these homologous chromosomes are going to make copies of itself during the S phase of the cell cycle. Remember G1, S, G2M, and the S phase is our DNA replication. So all these DNA are going to be replicated. So now you're going to have 2n equals to 12. But if there is non-disjunction happening during the mitotic process, you're going to have 4n. So instead of have 2n equals to 12, you're going to really have 4n, because for each type of chromosome now you're going to have 4 because they were never separated. The sister chromatids were never separated. So if this cell were to go through meiosis, you're going to have the original 2n cells again. And then if you have 2 of these 2n gametes, originally you're supposed to have 1n gametes, right? Because gametes supposed to be haploid or 1n. But if you have 2 of the 2n gametes coming together to make a new species, this new species is going to have 4n, and now you have a different number of chromosomes compared to the original amount that it's supposed to have. And this new species, you know, could continue on and stay as a new species. So the next one that we can see is allopolyploid is having multiple sets of chromosomes derived from different species. So right here we have 2 polyploid gametes. So anytime you have gametes that's more than 1n, that's called a polyploid gamete. So these 2 all came from the same species. This is talking about 2 different species producing polyploid gametes, and that the combining of those 2 polyploid gametes can produce an offspring that eventually become a new species. These cannot interbreed with either parent species usually. So let's take a look at how this could work. You could have, and then this generally, it says allopolyploid usually can't mate with either parent species. This usually is because of the difference in chromosome numbers. They're just too different. It doesn't work. So let's see how this works. How this doesn't work, I guess. We have this one species of plants that have 2n equals to 6. That means there are 6 individual chromosomes or 3 pairs of chromosomes. And you have this other species of plants that has 2n equals to 4, meaning that there are 4 individual chromosomes. They look like they're attached, they're not. There are 4 individual chromosomes and there are 2 sets of chromosomes. So that's what usually happens with plants. Different types or different species of plants would generally have different numbers of chromosomes. So normally you would have gametes with n or haploid number equals to 3 and n equals to 2. You would if you were to put those two gametes together, there could be a zygote with n equals to 5. So what happens is, if this zygote, n equals to 5, this doesn't work, right? This one plant cannot go through meiosis at all because you can't cut n equals to 5 to half an n equals to 2.5. That just doesn't work. So this zygote is going to be sterile, meaning that it cannot reproduce. However, if there is another error, well, actually, so far, this is all normal, right? However, if there is an error causing, or a non-disjunction error causing the chromosome number to double, so instead of having your regular somatic cells as n equals to 5, you would have somatic cells as 2n equals to 10. This solves the whole problem. Earlier, we didn't have any pairs of chromosomes, but now we do have pairs of chromosomes. In fact, we have 5 pairs of chromosomes, and now 2n equals to 10, right? So in this case, this 2n equals to 10 can then go on through meiosis and allow n equals to 5, and then when you have n equals to 5 cells combining together, you will go back to 2n equals to 10. So it's all about the chromosome number. Can this chromosome number allow meiosis to happen, basically? So this one doesn't work because meiosis cannot happen to n equals to 5, but because of this error, now we have 2n equals to 10, and meiosis can happen, and now that means reproduction can also happen. So this is just an example. This actually, this whole polyploidy situation that we've been talking about the past too many minutes, this actually matters because there are actually a good amount of species that originated by polyploidy, through the process of polyploidy. These are only the ones that we know, and there are plenty more plants that we don't quite know about. So here's an example. These flowers, these tea whatever flowers, they arise from the combining of 2 different species, together. So n equals to 12, I suppose. N equals to 12. Well, 2n equals to 12. 2n equals to 12. So you have n equals to 6, n equals to 6, and you have together you will have 2n equals to 12. But that doesn't work yet because there's no pairs of chromosomes or homologous chromosome. Through non-disjunction, you would have 2n equals to what did I say? So this should be n equals to, 2n equals to 12, 2n equals to 12, n equals to 6, but because these chromosomes are different chromosomes, when they combine to form a zygote, you would have n equals to 12, not 2n equals to 12. I think that was the mistake I made. So this n equals to 12, if that goes through non-disjunction again, you could have 2n equals to 24, and this 2n equals to 24 can then go on to produce gametes and allow continued reproduction to allow these 2 hybrids that can survive and reproduce. This just shows you that this is not that important. All right, moving on to sexual selection. Sexual selection can also drive sympatric speciation because let's say you have a group of birds. They don't have to select certain types of birds, right? Through a certain mutation within the female birds, for example, they could naturally just, through their natural instinct, decide that certain male birds are more preferred compared to other male birds, even though all the birds, male and female, are living within the same area. So sexual selection of mates of different colors has likely contributed to speciation in cichlid fish in Lake Victoria. So here is the example that we have. These fish all live in the same area, and through the sexual selection process of female fish to male fish, as you can see, there are different patterns of these 2 types of fish as a result of this sexual selection process, and they became 2 different species. Now here's an abstract that I would like you to read. We might read it in class some other day, but basically this is the abstract of this particular experiment, and I think reading abstract, is helpful for your FRQ and multiple choice because a lot of the times the AP biology committee would look at a scientific paper and actually take out pieces of information to ask you a question. So we'll look at this some other time. Continue on. Habitat differentiation. Sympatric speciation can also result from the appearance of new ecological niches. So if you think about all of these organisms living in the same area, there could still be different niches. So if you don't know what a niche is basically where this population lives, how it interacts with the environment, how it interacts with each other, basically all the environment that goes into, this one population. The interaction between this one species with other species, between individuals within the same species, between the species and the environment, all of those are called ecological niches. So within the same area, there could be different niches that arise. So here's an example. Populations of the North American maggotsfly can live on native hawthorn trees as well as more recently introduced apple trees. And flies that use different host species experience both habitat and temporal isolation. So we have these two types of trees, and the new ecological niche is introduced because people introduced apple trees. So now you have these flies, these maggot flies, that either live on the native trees or on the apple trees. And the result of this is that, now they have this habitat isolation because they're on different trees, and there's also temporal isolation, I suppose, because these two types of trees produce their fruits at different times of the year. So let's take a look at, it doesn't show you. Anyway, there's no picture of this one, but I hope this kind of makes sense. Where'd it go? This right here. I hope it makes sense to you. There might have been a picture on your paper. I don't remember. So now here's a review for allopatric versus sympatric speciation. Allopatric speciation, geographical isolation, restrict gene flow. That's the most important part, right? This is all about allopatric speciation. Reproductive isolation may arise as a byproduct. It's not necessary, resulting from natural selection, genetic drift, or sexual selection. Actually, sexual selection could happen, to allopatric speciation as well, right? But of course, there should be mutation added on to this as well. Mutation is the origin of all of these possibilities. And then the thing about speciation is that even if the contact, or allopatric speciation is, even if the contact between the two populations are restored, that there's no longer geological barriers because there's so much differences accumulated, the interbreeding is still not going to happen. So all those pre and post zygotic isolation, mechanisms, at least one of those will prevent the two species from having actual gene flows and allowing them to go back to the same species. So now, sympatric speciation, you would have a reproductive barrier isolating a population without geographical separation. So you have the same gene pool, sort of. They live in the same area. However, there could still be reproductive barrier happening either through these three possibilities: polyploidy, natural selection, or sexual selection. That's it.